Okay. So the basis for, so the plasma membrane is universally described as being a selectively permeable membrane, okay? Meaning that some things can get through, some things can't get through, okay? And the basis for which things, how things can cross the membrane, the basis for that is the structure of the membrane, okay? That it is predominantly a lipid membrane. So in general, things that are hydrophobic, that are fat soluble, have a fairly easy time crossing the membrane, and things that are water soluble have a harder time passively moving across the membrane. Okay, that's the general rule. So the things that determine the ease with which something can cross the membrane is lipid solubility is number one. Okay, and then also size and what particular substance you're talking about. So things that are polar but small, okay? Things like CO2, for example. CO2 has a polar structure. CO2 has some degree of ease in crossing the plasma membrane, okay? Because the polarity is not as strong as some other polar structures. Oxygen is actually a fairly nonpolar structure, right? Molecular oxygen are two oxygen atoms double bonded to each other. So oxygen's going to be forced to share those electrons pretty equally. So that's a fairly nonpolar structure. So oxygen can get cross membranes very easily. Thank God, right? Fatty acids, they're fat soluble. They can cross membranes pretty easily. Small polar substances, okay? There are some passive channels in the membrane that allow small polar substances to cross, okay? And some of these protein channels are selective for certain things. So there actually are some passive channels that allow sodium to enter the cell, passive channels that allow potassium to leave the cell, okay? Passive channels that allow other charged particles to get in and out. But the density of these passive channels really depends on the cell type you're talking about. Okay? Big variety depending on what cell type you're talking about. And then water is a very particular situation. Okay? Water can generally freely cross the membrane, but it can only passively move across the membrane in limited amounts, okay? Because water actually has a fairly polar structure. So water can pass through the membrane in limited amounts, but most cells in our body have abundant what are called aquaporins, or water channels, that allow water to cross the membrane freely. Okay? And these aquaporins, this is actually the structure of a particular subtype of aquaporin. The different subunits are color-coded. Okay? So it's a 4-subunit structure. It sits in the membrane. So you notice that these, the part of the protein that's sitting in the membrane look like they're just a bunch of barrels, right? Those barrels represent those helices, right? Those alpha helix, that alpha helix structure. And when you have a protein that spans the plasma membrane, the majority of the portion of the protein that sits in the membrane tends to form multiple alpha helices. Okay? So talk about structure equaling function. Okay? So you have this very elaborate protein with these this hydrophobic core sitting in the membrane, and then it just forms a little hole, little channel that allows water to pass through. Okay? And aquaporins allow water to freely shift in or out of the cell. So generally speaking, unless you're talking about a very specific cell type, okay? And when we get to the renal system, we'll talk about cells in the nephron, that some cells, their plasma membranes don't have aquaporins. Okay? But your generic run-of-the-mill cell, plenty of aquaporins, and water can freely move in or out. So when substances are crossing the membrane, they tend to move either passively or they move actively. And when something moves passively, it doesn't require any input of energy. Right? It's analogous to something rolling down a hill. All it needs is opportunity. It doesn't need energy. It just needs opportunity to roll down the hill. Okay? When something is being transported actively, you have to put energy in because it's analogous to pushing the boulder uphill. Right? The boulder, even if it has opportunity, boulders don't spontaneously move uphill. Right? You got to really get your energy in there and really push that boulder up. Okay? So the same thing with crossing the membrane. So passive transport, or the movement sort of downhill, is in the form of either simple diffusion, facilitated diffusion, or osmosis. Okay? And then active forces, we've got two flavors of active transport. We have primary active transport, which is very straightforward. We're using ATP and we're moving something uphill. Okay? Secondary active transport, it's a little less straightforward. We're actually using a slightly non-intuitive form of energy, not ATP, to move something uphill. Okay? So passive forces, simple diffusion. Diffusion is that things are going to naturally tend to move from an area of high concentration to an area of low concentration. So molecules that are more tightly packed together, they're going to collide into each other, and then slowly they're going to take up more space. They're going to diffuse into the area of lower concentration. So you've got two sides of a container separated by a semi-permeable membrane. Okay? The Brownian motion here is creating a tendency towards movement to the right, and the Brownian motion here is going to create a tendency for movement to the left. But since the concentration on the right is so low, that movement to the left is not very much, right? It's certainly not compared to the movement to the right. So you get a net diffusion to the right until you get an equal distribution on both sides. Okay? So now the tendency for movement in each direction is equal. So diffusion requires a concentration gradient. Okay? And it's always trying to get to equal on both sides. Okay? So that's the crux of diffusion. And diffusion, the rate of diffusion can be altered or increased or determined by a variety of things. So there's something called Fick's law. Very smart man, Mr. Fick. He came up with several factors that are going to determine the rate of diffusion. The first is the magnitude of the concentration gradient. Okay? The higher the high is compared to the low, the steeper the gradient, the faster the rate of diffusion. Okay? And as that gradient, as the diffusion proceeds and that gradient starts to get smaller, the rate of diffusion starts to go down. Okay? Another one is the permeability of the membrane. Okay? As if you have a more permeable membrane, you have faster rate of diffusion. Third is the size or weight of the substance diffusing. Something that's smaller, lighter, is going to diffuse faster. The next is the thickness of the diffusion gradient. The thinner, or the thinness of the diffusion gradient. The thinner the diffusion, not gradient, the diffusion barrier, excuse me. Okay? The thinner the diffusion barrier is, the faster the rate of diffusion. Okay? Thinner barrier, it gets across faster. This one right here.
And has that difference in the permeability of the membrane?
It's different because you can have two membranes that are of equal thickness. One can be more permeable to the substance. It's going to diffuse faster compared to the other. They're very similar, but slightly different. Another way of saying the thickness of the barrier is also the diffusion distance. Okay? Whether it's a barrier or it's just distance. Okay? And that becomes very important because you'd be surprised how many absolutely critical processes to survival are accomplished through simple diffusion. Okay? How does oxygen get from your lungs into your blood? Diffusion. How does oxygen get from your blood into your tissues? Diffusion. Okay? So at those sites of oxygen exchange, you'll find a very, very small diffusion distance, very thin barrier, very, very permeable. Okay? You'll find the concentration gradient to be fairly high. And this last one is a huge mechanism for maximizing diffusion in the body. This is the surface area available for diffusion. So you can see we've got this barrier, but not all of it is available for diffusion. It's just this one little area. The greater the surface area available for diffusion, the faster diffusion happens. Okay? And so everywhere in the body where diffusion, simple diffusion is the primary mechanism for movement, you see this tremendous maximization of surface area. Okay? And we talk about that in the lungs, we talk about that in the capillary beds, we talk about that in the GI tract with absorption of food. Okay? And in the case of carrier proteins, the protein structure is such that the binding site for the substance is either facing one direction or facing potentially both directions, but it's only going to carry the substance from an area of high to low. So this protein is sitting in the membrane. When the substance binds at the binding site, it changes the structure of the protein. Okay? And it changes the structure of the protein so that the protein is now facing the other side. And as soon as that structural change happens, it loses its affinity for the substance. And so the substance is let go. And as soon as it lets it go, it goes back to its original shape. Okay? So with carrier proteins, the only thing that's necessary is that there's a gradient, there's a concentration gradient, and that the substance has to bind to the binding site. So it's still simple diffusion. So an example of a substance that crosses most membranes this way is glucose. Okay? Glucose transporters are facilitated diffusion carriers. They bind to glucose and they carry glucose across the membrane down its concentration gradient. Okay? Yes, ma'am. What do you mean? It's more a function of probability, right? That the likelihood of binding is greater at the end with the higher concentration. The likelihood that the substance binds to the protein, yeah, is higher in the area of higher concentration. Yeah, absolutely. So if the concentration gradient switches, okay, many of these protein carriers will change the direction of movement. Okay, yeah, so some of them are like revolving doors that will move in either direction depending on the concentration gradient. Okay, so question to you. This slide, I've got this graph. I've got on the y-axis, I've got rate of diffusion. So going from 0 to increasing rates of diffusion. And on the x-axis, I've got concentration of substance that's being transported. Okay? Going from 0 to increasing concentrations of substance being transported. So for something that is crossing the membrane, if it can cross the membrane, you would expect that as I increase the concentration on one side, that the rate of diffusion will increase, right? You agree? So as I increase the concentration, let's say on the outside of the cell, that the rate at which that substance moves in the cell is going to increase. Now, there are two transport mechanisms represented by two different lines. In the black line, that's the relationship that's being drawn, right? And it's just a straight line moving off into infinity. That as you increase the concentration, the rate of diffusion increases. But in the green line, starting at 0, as you increase the concentration, the rate increases until it hits some limiting factor, and the rate of diffusion is actually plateauing. So one of these represents simple diffusion, and one represents facilitated. Which one represents simple diffusion? Who thinks the black one? And who thinks the green? So the black line represents simple diffusion. Okay? That the more you increase the concentration, the more it crosses. Okay? The more transport. And the faster the rate of transport. On the other hand, facilitated diffusion is being represented by the green line, meaning that as the concentration of the substance increases, the rate of diffusion also increases, but then at some point you hit a limiting, you hit a really a upper ceiling in terms of rate of diffusion. What's happening?
There's a limited number of protein channels.
There's a limited number of protein channels that can carry this substance across. Okay? So this red line represents the maximal rate of diffusion that is going to be determined by the available transporters. At some point, you're going to saturate your transporters. All your transporters are running at maximum speed. They're all occupied, and you can't transport the substance any faster. So that is an important differentiating feature of facilitated diffusion versus simple diffusion. That you have a saturation point, okay? Because you have a limited number of transport proteins. Okay? The other feature, in addition to saturation, is specificity. Okay? that because the substance has to bind to a carrier protein, there is some specificity involved in that transport. Okay? You can't have any random substance binding to this protein. It's got to be that particular substance or something that's very similar to it. Okay? Does that make sense? Good. And when we talk about, this is actually something that becomes very clinically relevant when we get to the renal system and we talk about things like the reabsorption of glucose in the kidneys. So without getting too far into it, the way the kidneys work is we basically put everything that's in blood, anything that's dissolved in blood, minus the cells and the protein, we just dump that into the nephron, right? And then we start taking back everything we absolutely need, and everything else just gets eliminated in the urine, okay? So glucose, under normal circumstances, we take back all the glucose that was originally dumped into the nephrons. Okay? So that what's normal is that someone should not have glucose in their urine. But glucose is being transported by these transporters. So if someone has diabetes and their blood glucose quadruples, right? The concentration of glucose all of a sudden is too high. It maxes out the rate of transport, and you still have glucose trapped in the urine. Okay? So that becomes a real clinically relevant example to this difference in transport mechanisms. Yes, ma'am?
Do these carrier proteins carry across multiple kinds of substances, or are they particular to one kind of substance?
They are usually fairly specific, but everything has sort of a preference for one thing and then like a relative affinity for other things. So it's possible for some things to sort of like masquerade as glucose and get across, but it's usually that transporters are very specific to their particular transport substance. Yes, ma'am?
What's the concentration next to what's supposed to be the simple fusion?
Say that again?
What's the concentration to get to what they're supposed to be the simple fusion line?
So I see where you're trying to get, but what I'm doing is I'm constantly increasing the concentration. I'm maintaining that concentration gradient. That's what this is saying, right? That it's not like the previous slide where eventually it's going to equal out, because what it would be like is if I constantly added more here, right? Then you would have this continuous movement. Okay. Osmosis. Osmosis is, I'm gonna move on. I see your hands, so I'll solicit some questions in just a little bit. So osmosis is actually a form of simple diffusion. Okay? Osmosis is a form of simple diffusion, but it involves the movement of water across a semi-permeable membrane. Okay. And osmosis is one of these like deceptively simple concepts, right? That you think that you fully understand, and then like you get into it and it's like, oh, that's really interesting. It's more complicated than I thought. So the key features with osmosis is that you have two fluid compartments. You've got a semi-permeable membrane in between. Okay? And the two fluid compartments are going to contain solutes. They're solutions. It's not pure water. They're solutions, and some of those solutes, this is critical, some of those solutes cannot cross the semi-permeable membrane. Okay? Osmotic pressure can only be exerted by a solute that cannot cross a membrane. Okay? osmotic pressure can only be exerted by a solute that cannot cross the membrane. So if we're looking at the left side of the slide, so that we've got side 1 and side 2, if we look at these dark blue circles, we've got the light blue circles and the dark blue circles. The light blue circles can cross the membrane. Okay? Because they can cross the membrane, they're going to evenly distribute. They're going to diffuse, and then they're going to evenly distribute. Okay? No osmotic pressure. The dark blue circles are impermeable to the membrane, so they can't evenly distribute. Okay? So now you have two compartments with different concentrations. Okay? That's not gonna fly. All right? Because what water is going to do is, if the solutes can't move, then the water is going to move in order to equal the concentrations. But in order to equal the concentrations, it's got to get to different volumes. Does that make sense? In order for, so here on the side 1, we're starting off, we have 2 dark blue circles. On side 2, we've got 4 dark blue circles. The concentration of dark blue circles is higher in side 2. Okay? Another way of thinking about this is the water concentration is higher in side 1. Right? There is more water to solute in side 1. So in order to equal the concentrations, water has to move from side 1 to side 2 so that the solutes per volume, that's what concentration means, solutes per volume, unit of volume, is equal on both sides because the volumes are different. That make sense? So when we talk about osmotic pressure, these solutes on side 2, they exert an osmotic pressure. And osmotic pressures are pulling pressures. They pull water towards them. Okay? They pull water towards them. So these solutes that are impermeable to the membrane on side 2, because they are of higher number on side 2 per volume, they're going to pull water towards them until you have equal concentrations on both sides. All right? So where osmosis becomes very important, so there's a couple of terms. Osmosis is the phenomenon, right? And a way of thinking about osmosis, because it is analogous to simple diffusion. So simple diffusion, a substance was moving from high to low concentration, right? With osmosis, I know this isn't quite right, but you can think about it this way, is water is moving from an area of high water concentration to low water concentration, okay? That water is equaling out in concentration.
So in the second, on the right-hand side, is there still osmotic pressure being exerted in that instance, or no?
Once it hits this level, there's no more pressure, yeah, because now they are equal in concentration. Okay? So where this becomes very important is actually when we talk about tonicity, clinically, when we talk about tonicity, because tonicity is the effect that a solution will have on cell volume. Okay? The term tonicity describes it's a property of a solution, and it describes the effect that that solution will have on cell volume. Okay? So, and things are described as being either isotonic. So if a solution is isotonic, iso means same. Okay? It has the same Osmolarity, osmolality, those terms are interchangeable to a great extent. It has the same osmolarity as the inside of a cell. An isotonic solution has the same osmolarity as the inside of the cell. Okay? As the same solute concentration. So what direction is water going to move? It's not, right? So quick drawings. The osmolarity of a cell is approximately 290, and the units on osmolarity are what's called milliosmoles. Okay? And osmolarity is actually a measure of the number of particles in a solution that can exert an osmotic pressure. Okay? The number of particles in a solution that can exert an osmotic pressure. An intracellular fluid has an osmolarity of about 290. So if this cell is bathed in a solution that is isotonic, what is the osmolarity of this solution? 290. Okay? No net movement of water. Okay? If the solution, on the other hand, is hypotonic. Hypo meaning less than, right? If it's hypotonic, it means that a hypotonic solution is a more dilute solution compared to intracellular fluid. It has fewer particles. So what's the number in relation to 290? Less than. Okay? So let's say it's 280. And this is something, when we get to the renal physiology, this becomes, again, a big deal, that as the numbers get smaller, the solution gets more dilute. Okay? More water, less particles. So this hypotonic solution is going to cause water to shift. In what direction? Out of the cell or into the cell? Into the cell. So hypotonic solutions cause cell swelling. Okay? Hypotonic solutions causes cell swelling. It causes this water to move into the cell. The cell gets big and bloated. Okay? Hypertonic solutions have an osmolarity greater than the cell. So let's say it's 295. The higher the number, the more concentrated the solution. Less water. What direction is water moving? Out of the cell. Hypertonic solutions causes cell shrinking. Okay? So where this becomes extremely important clinically? Where are my nurses? IV fluids, right? IV fluids, extremely important. And there's a wide variety of standard IV solutions. We've got the variety of isotonic solutions. What are some of the isotonic solutions, guys? Normal saline, lactate ringers, right? And even there's like the sort of isotonic, D5W, right? Dextrose water 5%. The reason why it's sort of isotonic, so dextrose water is basically sugar water. And it has the same osmolarity, but it's dextrose. It's a sugar. And the reason why it's sort of isotonic is because it goes in isotonic, but then what happens when you start absorbing that sugar and you start metabolizing that sugar? All of a sudden that solution becomes what? Hypotonic. Okay, because your particles are going away. Okay, so you've got normal saline, which is what, 0.9, I believe? Yeah. Normal saline, Lactate ringers is a isotonic solution, and then D5W is an isotonic solution. So, and then you also have a variety of hypertonic solutions and hypotonic solutions that are appropriate depending on the clinical situation. But tonicity of solutions become extremely important when you're talking about IV fluids. Okay? Yes, ma'am.
When you were talking about the C5Ws, is that, and you said that you absorb the sugar in it, does that happen every time you use it, or is it just the some?
No, you're going to absorb it no matter who you are, no matter what the situation, yeah.
It's only like isotonic.
Exactly, exactly. But there are situations where D5W is the appropriate solution to use. Yeah. Okay. Moving on. Okay, so active transport. Active transport, primary active transport, secondary active transport. Okay? Primary active transport, very straightforward. It uses the energy in ATP. Okay? And ATP is hydrolyzed, so it goes from adenosine triphosphate, so adenosine and 3 phosphates, to 1 phosphate comes off, and it's adenosine 2 phosphates plus 1. Okay? And that breaking of that bond releases energy, and you can do work with it. So probably the most important example of primary active transporter is the sodium-potassium pump, also known as sodium-potassium ATPase pump. And this ATPase refers to one of the subunits in the transporter. Sorry. That's gonna be great on the recording. One of the subunits on that protein is an enzyme, and it's an enzyme that's dedicated to hydrolyzing that ATP. Usually enzymes usually are written out as ending in ASE, right? ACE. So it refers to the enzymatic subunit in that protein. So the sodium-potassium ATPase pump is found in every cell in our body, and it's constantly working. It's constantly working. Something like over 50%, depending on the cell type, but over 50% of the energy of the cell is dedicated to running the sodium-potassium pumps. Okay? The sodium-potassium pumps, it pumps 3 potassiums out, 3 sodiums out, and then pulls 2 potassiums in. Okay? In one sort of go around, it kicks out 3 sodiums and pulls in 2 potassiums. And both of those ions are moving against the gradient. Okay? So this pump is absolutely critical for maintaining the ion concentration gradients between inside and out of sodium and potassium. Okay? So very important. Another form of active transport is vesicular transport, and I'm not going to talk too much about vesicular transport, but basically it is the movement of a substance into the cell or out of the cell through the formation or the incorporation of transport vesicles. So when a substance moves from inside the cell out of the cell, that process is called exocytosis. Okay? So let's say you are synthesizing a protein that, let's say the protein is a hormone, right? This is an endocrine organ. It's synthesizing its hormone, and it gets a signal from the outside to release that hormone. So that hormone is being stored in secretory vesicles, and they're just little bubbles of plasma membrane that contain something. Okay? That's what those vesicles are, little bubbles of plasma membrane. So this bubble of plasma membrane filled with hormone moves to the plasma membrane, and then the vesicle opens and it just incorporates into the plasma membrane and just dumps out the hormone. That's exocytosis. Okay? Something moves to the surface, the vesicle moves to the surface, incorporates into the membrane, and gets released. The opposite process is endocytosis, and that's when a substance that is in the outside, the extracellular space, gets internalized, right, and enters the cell by forming a vesicle and internalizing the vesicle. Okay? Now, endocytosis can happen in a variety of circumstances. It can involve a receptor, so the substance could bind to a receptor and then the entire thing gets internalized, or it can happen without the binding to a receptor. Okay, so it could be receptor mediated, receptor non-mediated. I'm not going to get into the details, but basically vesicular transport is another important example of primary active transport. Involves ATP. Secondary active transport is using energy, but it's not energy in the form of ATP. Okay? Secondary active transport is using the energy that is stored in the concentration gradients, predominantly the ion concentration gradients across the membrane. Okay? So what that means, let's take sodium. Sodium is an important one. Okay? Sodium has a higher concentration where? Outside the cell, right? So sodium has a higher concentration outside the cell, which means that it has a big motivation to get where? Inside the cell. Okay? So as long as sodium is sitting outside the cell and wants to get inside the cell, it's very similar to a boulder sitting at the top of a hill with a little wedge keeping it from coming down. Okay? So that's the little sodium boulder, and it really wants to go down. So the fact that it is in this position means that it has a lot of what we call potential energy. Okay? Now, let's say I take the wedge away. What happens to this? Sorry. Okay. Back to the boulder. I take the wedge away. What happens to that boulder? Starts screaming down that hill. All the potential energy gets transferred into what kind of energy? Kinetic. As all that energy is getting released into kinetic energy, if I was smart, and if I was smarter than I currently am, and could engineer something that could harness that kinetic energy, I could do work, right? And this is actually the basis for like hydroelectric power. You know, so you build like the Hoover Dam, and so all of this potential energy is jammed up against that little thumbnail of a dam. And then I just like open up a little spout and all this water comes rushing out. Okay? And I've engineered all of that I've engineered something that converts that movement of water out. It turns turbines and that creates electricity. Okay? So what secondary active transport is, you have these transporters that harness the kinetic energy released when sodium moves into the cell or some other ion moves down its concentration gradient, and it uses it to actively move a second substance against its transport against its concentration gradient. So there's a lot of examples of these, and many of them use sodium because the sodium concentration gradient is significant and it's very well maintained. So sodium moves into the cell, and we can actively pump then amino acids into the cell, right, against its concentration gradient. Or we can move calcium out of the cell. Okay? But the nature of the structure of this protein is what's actually engineering the harnessing of that energy and the conversion of it into something that does work. Okay? So in secondary active transport, there's always two substances moving. There's always at least two substances moving. One of them is moving downhill in terms of its concentration gradient. And the other substance is moving uphill. Okay? So secondary active transport, there's always at least two substances moving. One is like the helper substance, like sodium, and the other is the actual substance of transport. All right? So examples. So no matter what's happening, these two substances are always moving in opposite directions with respect to their concentration gradient. Okay? So you might want to write that down. That's important. The two substances with secondary active transport, they are always moving in opposite directions with respect to their concentration gradients. Okay? With that said, if you're looking at whether they're moving into the cell or out of the cell, sometimes they're both moving in, sometimes they're both moving out, and sometimes they're moving in different directions. Okay? So when two substances are moving in the same direction with respect to extracellular and intracellular movement, we call those co-transporters. Okay? So these are examples of co-transporters. And if you notice, there's a lot of them that use sodium. Bless you. That's not actually what you say when somebody hiccups, but so a lot of them involve sodium. So in every single example of these co-transporters, what direction is sodium moving in? Into the cell, right? Because that's downhill for the cell, for sodium. That's the direction of movement. And sodium is a great workhorse. That's what it does. It moves into the cell, it frees up energy. But for the rest of these that are also moving into the cell, that just happens to be the... Is that a baseball game? All right. I can't compete with a baseball game. So for these co-transporters that are using sodium, we know sodium is the workhorse, and we know sodium is moving into the cell. The other substance that's moving into the cell is moving against its concentration gradient. But its concentration gradient happens to against the concentration gradient happens to also be pointing inside the cell. Yeah. To their concentration gradient, that's absolutely right. So two substances, they're always moving in opposite directions with respect to their concentration gradient. One's moving downhill, the other one's moving uphill. Yes, that's what I meant. Yeah. That they're moving in opposite directions with respect to each other. So one is moving uphill, the other one's moving downhill. Yeah. Okay. Um, co-transporters, they're both moving into the cell or they're both moving out of the cell. With exchangers, I know it makes no sense right now. Believe me, when you're not at, I promise you when you wake up in the morning it will make more sense. You've been in class for like 13 hours or something crazy, all right? Exchangers, they are moving in opposite directions with respect to into the cell or out of the cell, all right? So we have this interdependence between the cells that really are the basic units, the tissue types that they form, the organs and the organ systems, okay, that maintain the health of the organism as a whole, but then that organization in turn maintains the health of the cell. So throughout the semester, we cover some really key organ systems of the body. We will cover the digestive system, the cardiovascular system, the respiratory system. And we cover these two systems, this nervous system and the endocrine system. We actually talk about those systems first because those two systems in the body are our major control systems. They are responsible for regulating, controlling, coordinating the function of all of the other body systems. So we talk about them first as a way to understand how overall function of the body is controlled. Last time when the lecture last time, we talked about some of these key foundational concepts of physiology, but we ended with the idea of homeostasis and homeostatic control mechanisms, right? And our example homeostatic control mechanism was body temperature regulation, right? Thermoregulation. So a major homeostatic regulation system like thermoregulation is going to be something that is governed and controlled by either the nervous system or the endocrine system or both. Okay? Now, these two control systems have many similarities and many very important differences. The most obvious difference is in their anatomical arrangement. The nervous system is a hardwired system. There are physical connections between all the elements of the nervous system. Okay, so there are physical connections between all the elements of the nervous system. The endocrine system is an entirely wireless system. Okay, it's composed of all of these endocrine glands throughout the body that release hormones into the bloodstream. Those hormones travel through the bloodstream, and then they can act on multiple targets throughout the body. And hormones basically act, when they are released by an endocrine gland, they will act on all organs that happen to have a receptor that can bind for that hormone. So by doing that, you can end up with 6, 7 different effects exerted by a single hormone. So wired system versus wireless. The type of chemical messengers, the nervous system and the endocrine system both use a chemical messenger. Because of the pattern of their release and where they act, they are given 2 different names. Okay? We call chemical messengers released by the nervous system neurotransmitters. Okay? By and large, we call them neurotransmitters. And a neurotransmitter is defined by how that chemical messenger is released. Okay? It's released by a neuronal cell, and it acts on a neighboring cell. Okay, so it only is released in small amounts, it diffuses a short distance, and it acts on a neighboring cell. Okay, that neighboring cell could be another neuronal cell, or it can be a target cell. The endocrine system releases what we call hormones. And a hormone is defined, again, by the pattern of its release. A hormone can be released by any cell. A hormone can be released by any cell, but it is released directly into the bloodstream. And therefore, it can act on a distant site. Okay? So you can have a neuronal cell release a hormone. And you can have any cell in the body release a hormone. When it's releasing a hormone, it is acting as an endocrine gland, even if it also happens to be, let's say, cells of the kidney. Okay? So a hormone and neurotransmitter both are defined by their pattern of release. Distance of action. Neurotransmitters act on short distances. Okay? They're released onto the neighboring cell. Hormones act on long distances. They travel through the bloodstream and they act at distant sites. Both have specificity in terms of what they will act on. And the specificity is based on the presence of a receptor that binds to the neurotransmitter or to the hormone. Okay, so both are going to act with a great degree of specificity. In terms of the speed of the response, okay, so how fast can you have the release of a neurotransmitter or a hormone and then its effect on the target cell? When it comes to the nervous system, this is a very, very rapid response. It happens within milliseconds, very, 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 very rapid. The endocrine system, it's a more delayed response. It's a longer time to get that response. We're talking minutes to hours to really see a true response. However, if we look at the duration of the action, how long that action is going to last, with the nervous system, it's fast on, fast off, usually. Okay? You can get a very, very rapid onset of a response, but usually that response is very short-lived. Okay? It's a very precise and very rapid action, fast on, fast off. The endocrine system, it takes a little bit longer to kick in, but it's a much more prolonged duration of action. And once that hormone is in the bloodstream, it's going to act on target cells until that hormone is eliminated by the body. Okay? And it's going to be eliminated by the body through a combination of the liver and urinary elimination. Okay? When an endocrine gland releases a hormone into your bloodstream, the kinetics of that action in terms of the how long it takes to get a response, how big that response is, and how long that response lives, okay? That kind of hormone action is very similar to how drugs work in the body. Okay? It's very similar to the pharmacokinetics of how drugs work in the body. Okay? We consume a drug, it enters our bloodstream, blood levels rise, and then they fall based on the rate of elimination. It's the same sort of concept. Okay? Except when we take a drug, it's something that we're introducing from the outside. When we release a hormone, it's something our body is doing from the inside. Okay? Both systems control all major functions of the body, and all have influence on the other control system. So the nervous system has controlled parts of the endocrine system, and the endocrine system controls parts of the nervous system. Okay, so very interrelated. So first we're going to talk about the nervous system. That's going to probably take us maybe 2 lectures, and then we'll talk about the endocrine system. That might take us another 2 lectures. Okay? Give or take. Okay, so talking about the nervous system, let's talk about the general organization of the nervous system. 2 major divisions of the nervous system, central versus peripheral. Central nervous system is comprised of the brain and the spinal cord. Okay. Central nervous system is brain and the spinal cord, and peripheral nervous system branches out from the central nervous system. Okay. And the peripheral nervous system branches out from the central nervous system, starting with the cranial nerves and the spinal nerves. Okay. And those cranial nerves and spinal nerves then branch into all of the nerves, all the peripheral nerves of the body. And there are 2 major types of nerves within the peripheral nervous system. And they are afferent nerves and efferent nerves. An afferent nerve is basically a sensory nerve. Okay? Afferent is synonymous with sensory. It's conveying some information about the body toward the central nervous system. An efferent nerve is some sort of effector or command or motor nerve. It is conveying some sort of command or effect that originates in the central nervous system and then goes out to the body. Okay, so afferent is sensory, efferent is command or motor. Now, if we look at those two divisions a little more closely, in the sensory arm of the peripheral nervous system, we basically have two forms of sensory stimuli. Okay. We have what are called somatosensory stimuli, which on this slide is just called sensory stimuli. Okay. These are our somatosensations. These are sensations that we are generally very aware of, we're conscious of. Okay. They include our special senses. They include our sense of taste, smell, sight, taste, smell, sight. What are our other senses? Hearing, right? Touch, right? So all of those, right? So touch, sight, hearing, smell, taste. Okay, those are our special senses. On top of that, we have a whole array, and then touch, of course, includes a whole array of sensations, right? Within the sense of touch, we also have things like pain, we have things like temperature, pressure. Okay, so there's a whole array of sensations involved in touch. On top of that, we also have sensations that we're aware of, but we may not be aware that we're aware of. And one of those is what we call proprioception. And proprioception is the idea that you are aware of your body position in space. If you close your eyes, you still know that you're sitting in a chair. Right? If you close your eyes and I flip you upside down, you will know that you're upside down. Okay, that's what proprioception is. Yeah. No problem. Proprioception. I'm a bad speller. Ception. Can you see that? Yeah. No problem. So our somatosensory stimuli are sources of stimulation, sensory stimulation, that we are consciously aware of. Okay. It's sensory stimulation that we are consciously aware of, somatosensory stimuli. And that is very different from our visceral stimuli. Visceral or viscera refers to our internal organs. Visceral stimulation are sources of sensory stimulation that we're not consciously aware of, but that sensory information is constantly fed to the brain, and it is designed to inform the brain on the status of body function, things like blood pressure, heart rate, okay? Plasma glucose, an array of sources of information. Okay? But they are information that's based on visceral function, baseline function of the body. This visceral stimulation is conveyed to the brain through our autonomic nerves. We'll talk about the autonomic nervous system in the efferent division in just a moment. So this is the sensory stimulation coming into the central nervous system, coming into the spinal cord and brain. And then the efferent division, which is the command division coming out from the central nervous system to the body. Okay. Again, there are two divisions. There are divisions that we call the somatic nervous system or somatic efferent division. Okay. And then we have our autonomic nervous system. So the somatic nervous system are comprised of our motor neurons, our motor nerves. Motor nerves innervate skeletal muscle. This is the arm of our efferent division that we voluntarily control. This is the arm that we voluntarily control. So when, for example, you put your hand on the stove and you feel pain. Put your hand on the stove and you feel pain. That sensory stimulation is somatosensory, right? It's related to your sense of touch. That information is conveyed to the brain, and the brain does something about it by you consciously removing your limb, right? You contract your skeletal muscles and you voluntarily move your hand off the stove. And it happens so quickly, it happens incredibly quickly, but as soon as your brain realizes that it's feeling pain, it moves your hand off the stove. The autonomic nervous system are commands that you don't control, you don't voluntarily control. They are under unconscious control, involuntary control, and they regulate visceral function. The autonomic nervous system regulates visceral function. Okay. The autonomic nervous system increases or decreases your blood pressure, increases or decreases your heart rate. Okay. Your autonomic nervous system stimulates the release of a hormone to regulate your blood glucose, and a number of other visceral functions. Okay. There are two divisions within the autonomic nervous system, so still under involuntary control. Two divisions. We have the sympathetic nervous system and the parasympathetic system. Okay. Under most conditions, your body, your visceral function is governed by the parasympathetic nervous system. The parasympathetic system controls baseline function. Sometimes it's called rest and digest. It governs the resting conditions of your body. And then in specific situations, okay, when your body is in a position of being stressed or you have to mobilize a lot of energy, you get the dominance of the sympathetic nervous system, which is our fight or flight response.